Hello everyone, welcome back. So before we start our discussion on meiosis, we'll just look on to these two terminologies, which is centromere and kinetochore. So these are the two structures that is seen on the chromosome during the process of the division during the M phase, either mitosis or meiosis. So we'll see the first one, which is centromere. So if I take a chromosome like this, centromere is this particular region or the middle region of the particular chromosome. So if you take the sister chromatids as separate, if you separate the sister chromatids into two, so this will be the point, and this particular region which is loosely compacted, the other regions are highly or tightly compacted, and the in this particular region I have shown it as a middle. It might not be always middle, it can be at different positions, like it can be like this. In this it is now at the lower position, centromere. In this case it is at the upper position. So the centromere can take different positions. So for our discussion now I'm taking it as a middle portion. So centromere is a region where the particular chromatin is loosely packed than the rest. That is why you are seeing you are seeing it as a compressed or a lightly narrow region in the chromosome, because it is not tightly compacted than the other regions. So this centromere is the region where, or this is the particular region where in every chromosome you can see such regions, and these regions are the places where you can see repeated sequences in your DNA, or they are called as repetitive sequences. Repetitive sequences are nothing but, if you write the sequence of a DNA, the sequence of the DNA is nothing but the order of the nitrogen bases. That means in the first nucleotide, if it is adenine, you write it as adenine. Then if it is thymine, you write it as T. Then if it is adenine, again adenine, then guanine, cytosine, etc. So you will have the complementary in the other one. I am writing only in the one half. So what happens is, in a long stretch, if you see the this particular sequences, in some instances or at some places, they will show repetitive sequences. Repetitive sequence, for example, the example of a repetitive sequence, if a particular sequence is being shown or seen as repeating in a particular stretch of DNA, repeating in the sense that if this particular GATC, this is a sequence, and in a particular area you can see GATC, GATC, etc., etc., 100 types. So GATC, that particular sequence is repeated for 100 or 1000 times. This is in one strand, so the complementary will be present in the other strand here. So if in this case this GATC is being repeated for say, or 100 or 1000 times, so such places where certain sequences are so everywhere it will not be GATC. I am taking it as just only as an example. So the kinetochore is a region in the chromosome where you can see such repetitive DNA sequences. So the place where repetitive DNA sequences are present in the DNA or the chromosome is called as a centromere. That's why I said the position may differ. So in every chromosome, so if you take in the case of humans, chromosome number 1 and chromosome number 21, the length of both will be different. Length will be different, the sequence will be different, and also the repetitive sequences, if it will not be in the same position. If it is in the same position, you will have at the same position. If it is in the center, they will have at the center. But for every so that means in homologous chromosomes, you will have at the same position. In both the homolog, the centromere will be in the same location, but for non-homologous chromosome it will be at different places, as I showed you earlier. It might be in the upper end, it might be at the lower end. So depending upon the where the repetitive sequences are present, centromere might take different locations. So centromere is the region in the chromosome where repetitive sequences are present, and another feature is it is the region where the chromatin is loosely arranged. It is having compaction. You don't take it as it is a region where naked DNA can be seen. There are histoproteins attached. It is in the form of a solenoid, but not in the form that is seen in the upper end. The upper end is more compacted than a solenoid for becoming a chromosome. In the other area where the centromere, it will be loosely arranged only in the form of a solenoid. No more coiling upper in the upper type than that of the solenoid can be seen there. As a result, you can see it as a particular narrow region in every chromosome. And this is the region, if you are staining the chromosomes, you can see this, the other regions in the sense that if you take the chromosome like this two regions will be darkly stained because they are highly condensed. This the region, centromere region, will be usually lightly stained because they are not more compacted than the other one. And this centromere region is also site for a protein during the process of mitosis or meiosis. This is the region where a particular protein binds to it. So centromere is nothing but it is a region of the chromosome. Kinetochore is a protein. Kinetochore is a protein which binds to the centromere region. So after a chromosome is or chromosome become the solenoid is becoming condensed into a form of a solid into a chromosome, and whenever the centromere region can be seen, what happens is, so only in this particular stage the kinetochore binds. The kinetochore will bind to the centromere region, and it is the kinetochoral region which is the binding site of the spindle fibers. So the spindle fibers binds to this kinetochore region. So these are the two structures that we should be discussing during the discussion of this particular mitosis or meiosis. So we are now straight away moving to the division which is called as meiosis. So as I said in the beginning, meiosis is the particular division that is restricted to the germ cells, and that results in the production of if it is in males, it results in the production of sperm, and in females it results in the production of the egg. In this particular division, as I said, in meiosis you have two nuclear division. Because it have two nuclear division, uh, this particular division is divided into two phases as meiosis 1 and meiosis 2. In meiosis 1, you have the nuclear division followed by a cytokinesis, and the other one which is meiosis 2. Meiosis 2 is nothing but it is exactly similar to the process of mitosis. That means the end product of meiosis 1 will undergo meiosis 2. So meiosis 2, the events are similar to that of mitosis in the sense that what is the result of the meiosis 1? That will be the type of parent cell which is involved in meiosis 2. So it will give you the cells, or the result of M2 will be daughter cells which is similar to the cells which is resultant with the M1. So that is similar to mitosis. So what we discussed in mitosis will be same as that of meiosis 2, mainly the spindle arrangement and segregation. Okay, that will be similar. That means what is the number of chromosome in the cell which is starting meiosis 2? You will get two daughter cells from that with the same number of chromosome. Because of the special type of alignment, the straight line alignment, spindle assembly, and separation. So we'll discuss this particular division called as meiosis. So in both the cases, you have that means which is happening both in the testis as well as the ovary. You have the stem cells which are called as spermatogonial cells. They are dividing, so they are dividing by mitosis to increase their number. As a result, you get many spermatogonial cells which will be dividing and dividing to get more number of sperms as well as the egg. So what happens here is, uh, we'll be discussing only with this particular one, which is called as, we'll discuss first with spermatogony, and then we'll move to the oogonial cells. In spermatogonial cells, one of the cell, it is now starting the process of meiosis. So meiosis 1. So in meiosis 1, what happens is, so in meiosis 1 is different from that of mitosis, in the sense that in meiosis 1, the important events of meiosis occurs, which is crossing over and recombination, which results in variation. If these two processes were absent, all of us would be like clones. It is these two events, which is recombination and crossing over, or crossing over and recombination, that resulted in the uniqueness among us. We all are unique, and that uniqueness is due to this particular process that is happening during the event of meiosis, both in egg and sperm, that is crossing over and recombination, which occurs during meiosis 1. In meiosis 1, two important events occur. As we said, meiosis 2 is similar to that of mitosis. The two events that only happens in meiosis occur in meiosis 1. One is, as I said, crossing over and recombination. Second one is reduction division. We said that meiosis is the one which is ensuring haploidy of an organism. That means the parent cell with 46 chromosome should give a cell with 23 chromosome, or a sperm with 23 chromosomes. So this reduction in chromosome number or reduction division occurs during the anaphase 1 of meiosis 1. Okay, and that is what we are going to discuss in detail here in meiosis, because meiosis 2, as it is similar, we'll not waste more time on that. We'll be discussing what is happening during the meiosis 1, the events in meiosis 1. So before moving on to the details of spermatogenesis and oogenesis, we'll see the events in during this particular process, which is called as meiosis 1. So meiosis 1 is also divided into 4 phases, which is suffixed with 1: prophase 1, metaphase 1, anaphase 1, and telophase 1. So prophase 1 is the particular phase in which the two events occur, which is recombination and crossing over, or crossing over and recombination. And as this particular events happen, and this occurs, and prophase 1 is more lengthier than the prophase of mitosis. For the sake of discussion, we are dividing prophase 1 into 5 different phases. They are leptotene, zygotene, pachytene, diplotene, and diakinesis. So what happens here is, it is during this particular phases the important events like crossing over and recombination occurs. So the main difference between the arrangement of chromosomes in mitosis and meiosis is that in mitosis we saw that every chromosomes were separate. They didn't pair up. Everyone were separate. As a result, they were aligned in the form of a straight line during mitosis. But here you can see the important process of pairing up. That means homologous chromosomes pair up during the prophase 1. You can see during the zygotene stage they are pairing up. So here you can see in this particular diagram also they have shown two homologous chromosomes are shown. So this is a bigger homologous chromosome. This is 2, and the other one is these two. And here you can see that these particular homologous chromosome, they pair up, and this pairing up is aided by the formation of a structure which is called as synaptonemal complex. That means protein bridges form between homologous chromosomes. So a homologous chromosome means it is a pair like this with 4 chromatids, and protein bridges form between these two homologous chromosome, and protein bridges form between homologous chromosome, and this it is between. So these two are the sister chromatids. Now the bridges are formed between non-sister chromatids. So bridges form between protein bridges called as synaptonemal complex forms between the non-sister chromatids of homologous chromosome, as you can see in this particular diagram. So they form between the non-homologous homologous chromosomes, and it is between the sister chromatids. Homologous in the sense that this is chromosome number 1, this is also chromosome number 1. And this particular event, it is occurring during the phase which is called as, please remember this particular phase which is called as Pachytene. The event called as crossing over and recombination occurred during this particular phase which is called as the Pachytene phase. In Pachytene phase, what happens is, after the formation of the protein bridges, the non-sister chromatid, they exchange the segments. They cross over, they recombine. That means they exchange their segments or exchange their DNA fragments. As a result, a new combination occurs, or a new combination is being formed, and that occurs, or that results in, and this particular events, crossing over and uh, recombination, result in what is called as variation among individuals. If these two processes were absent, there will not be variation. All the organisms which were sexually reproducing will be same. So these are the events, recombination and crossing over, that causes variation among individuals, and that particular processes that occur between non-sister chromatids of homologous chromosome. It occurs in the phase of prophase 1 called as Pachytene stage. Okay, after Pachytene, then comes the phase which is called as our important phase, which is called as the Metaphase 1. So here you can clearly point out, or clearly you can see the difference in arrangement. So difference in arrangement is, so here it is clearly shown here that, so if let me try to enlarge this. So here you can clearly see what is the difference in arrangement in metaphase of metaphase 1 of meiosis 1 and mitosis. So here, because the homologous chromosomes are seen in pairs, they align at the equatorial plane in pairs, not in the form of a straight line. Not every chromosome in the cell does not form a straight line. A straight line is formed, but in pairs. If this was a human cell, and if it was mitosis, you can see a straight line with 46 chromosomes. But in meiosis, you can see a straight line with 23 pairs, because they pair up. If you count, you can count 23 number, but in pairs, because they will be, because of recombination and crossing over, they are connected to each other. Okay, and because of this particular pairing up, what we discussed, or what we said, which was a criteria for mitosis to happen, which was, so straight line alignment is different. In that, individual chromosomes were aligning in the form of a straight line. In meiosis, here paired chromosome, as you see here, paired chromosomes are being aligned here. And other criteria was that every chromosome in the cell should receive spindle fibers from both the poles. That means if all 46 chromosome, if it is a human cell, it will receive, every chromosome is receiving the spindle fibers from both the poles. But the situation is different here. If you see it, you can see that this is a homologous pair, and in this homologous pair of chromosome, in this particular homologous pair of chromosome, you can see that only one of the homologous pair is receiving spindle fibers from one of the pole, and the other homologous pair receives from the opposite pole. So the situation is like this. So if I draw this as the homologous pair, spindle fibers from this pole is attached to this particular chromosome, and from the opposite side it is attached to this. If it was mitosis, this might have been the condition, and this might have been the spindle fiber. So here, this is the difference in metaphase 1, that is paired alignment. That means homologous chromosomes are aligned in the equatorial plane in pairs, And because of this paired arrangement, only one of the homologous pair receives spindle fiber from one pole, and the spindle fiber from opposite pole is received by the other homologous pair. As a result, what should happen in anaphase 1? If this was the case, we said that in mitosis, sister chromatids are being separated, because when the spindle fibers pull, the sister chromatids will get separated in mitosis. But what happens in this particular case? Here, because of this particular type of spindle assembly and paired alignment during metaphase plate, during anaphase, it is not sister chromatids, it is the homologous chromosomes gets separated during anaphase 1. In anaphase 1 of meiosis, homologous chromosomes separate into two different cells, and this segregation results in what is called as the reduction division. Okay, so now we'll see why this is called as reduction division. So take as an example, we said a cell with two chromosomes, take it as one homologous pair. So we are discussing a cell with only one homologous pair, and now it was attached like this. Okay, so the parent cell of this might have been like this with this set, and now after S phase etc., now it has reached here. Now it is M1 here. This was the homologous pair during M1. Now the spindle fibers are attached like this, and now during anaphase 1, what happens is, now this is going to be separated into two cells like this. Okay, one into this and other into this. So after meiosis 1, this will be the case. That means homologous chromosomes got separated. So imagine if it was our 46 chromosomes or our 23 pairs. So if it was 23 pairs will be aligned like this in pairs. As a result, one set of 23 will move to the other set of 23 will move to the other cell. As a result, you will be having 23 here and 23 here. So from 46 chromosome, what happened after meiosis 1? After meiosis 1, the chromosome number has reduced. So that is why you call, or you say that reduction division is happening during meiosis 1, and that too during anaphase 1. It is anaphase 1 which ensures reduction division. Reduction division is happening during anaphase 1. So now in the next case, what is meiosis 2? As we said, it is same as that of mitosis. So this particular, both the cells will undergo meiosis 2. We'll discuss in detail of one cell. So what happens is, I'm just drawing the anaphase and metaphase stage. So only one chromosome. So if it is 23, all the 23 will align in the form of a straight line like this, and they will be receiving spindle fibers from every in both the poles. As a result, sister chromatids will separate, and you will get two cells like this, each cell with 23 chromosomes. This also the same thing happens, each cell with 23 chromosomes. So either this might be, if it is happening inside the testis, it will result in the formation of an sperm, and if it is the case with the ovary, it will result in the formation of an egg, which is haploid. So this particular thing was meiosis 2, which is similar to that of mitosis, and this one was meiosis 1. So please keep in mind that this should be kept in mind. That is what is happening during anaphase 1 and what is happening during what is getting separated during anaphase of mitosis and anaphase of meiosis. To get a clear idea, you can see here. So this is an answer for why this paired alignment happens, and why sister chromatids are separated during mitosis, and why homologous chromosomes are separated during meiosis. The answer lies in this particular diagram. So the topmost, or this particular topmost one, it shows metaphase 1 and anaphase 1. So you can see, so first we will discuss about this particular two, which is same as that of mitosis, metaphase 2 and anaphase 2. So here you can see, when the chromosomes is aligned in the form of a straight line like this, the kinetochore, as I said, which is the site of binding of the spindle fibers, they are exposed on both the sides. Okay, as a result, the kinetochore, it is receiving spindle fibers from both the poles. As a result, the sister chromatids are getting segregated and separated and segregated into both the poles. So see here. So here you can see the difference in alignment. So here, when it is aligned in the form of pairs, they show the spindle fibers in one side. Okay, so you can see the difference. If it was like this, in this case, now this particular structure is aligning like this. That's the difference. And when it aligns like this, all the kinetochore will be here. In this case, it will be in both the sides. So it receives only this, it can receive only spindle fibers from a single pole. And now what happens is, so here you can see the color difference which is shown. This is what is called as recombination crossing over. As a result, they have exchanged their segments during prophase 1. So this is anaphase 1, where the homologous chromosomes separate, and that results in what is called as reduction division. Hope this is clear. So this is regarding the particular two, the main two differences between mitosis as well as meiosis. So in the next session, the introduction will be, we are concluding the introduction with the genetics introduction with the next session, where we'll see some more terminologies like the homologous chromosomes, then the genes, alleles, etc., which we require in the discussion of genetics.